Hi students, welcome to Smart Locus. In this video, we will see the chapter Anatomy of Flowering Plants. Anatomy. Plant anatomy includes nothing but the study of plant tissues. Plant tissues are nothing but group of cells. Plant tissues are broadly categorized into two based on their ability to divide. Actively dividing, meristem. Non-dividing, permanent. Then meristems are further categorized. Apical meristem, intercalary meristem, lateral meristem. Permanent tissues further categorized into two, simple and complex tissues. Simple tissues, example, parenchyma, collenchyma, sclerenchyma. Complex tissues, example, xylem, phloem. Components of xylem includes vessels, tracheids, xylem parenchyma, xylem fiber. Components of phloem include sieve tube, companion cell, phloem parenchyma, phloem fiber. So next let us see meristem in detail. Based on the position, meristems are broadly categorized into three: apical meristem, intercalary meristem and lateral meristem. First about apical meristem. Apical meristem is found at the apex, that is at the tips of both roots and shoot. Then what is the function? They are responsible for primary growth. They produce primary tissue, that is growth in length. Then tissues left behind from shoot apical meristem constitute axillary bud. Axillary bud later develops to form flowers or branches. The next about intercalary meristem. It's found in between mature tissues. And in grasses, it is found in grasses, it helps to regenerate parts removed by grazing herbivores. And intercalary meristems are also responsible for primary growth. So both of these, both apical meristem and intercalary meristems are primary meristems, responsible for primary growth of the plant body, growth in length. Lateral meristem, that is found in mature regions of both root and shoot, and they often produce a woody axis, and they are cylindrical meristems. A typical example for lateral meristem, it is also known as secondary meristem because they appear later. Typical example is cambium. Different types of cambium includes fascicular cambium, interfascicular cambium, cork cambium. Permanent tissues. Meristematic tissue undergo differentiation, that is structural and functional specialization to perform specific functions. And during this process, they loses the ability to divide and becomes permanent tissues. Permanent tissues are of two types: simple permanent tissue and complex permanent tissue. First, we will see about simple permanent tissue. Simple permanent tissue consists of only one type of cells. Three examples are parenchyma, collenchyma, sclerenchyma. Among the three, parenchyma and collenchyma are living tissues. Sclerenchyma is a dead tissue. First about parenchyma. Parenchyma is the major component in plant organs. So that otherwise we can say that it is the most abundant tissue in plant organs. And then the cells of parenchyma are isodiametric. Then different shapes. It can be either spherical or oval or round or polygonal or elongated in shape, the cells of parenchyma. Then parenchyma cells are thin-walled. That means cellulosic cell wall. Then the cells are either closely packed without intercellular spaces or loosely arranged with intercellular spaces. Then the functions in which parenchyma takes part are photosynthesis, storage and secretion. Next about collenchyma. Collenchyma, it forms hypodermis in dicot, that is a layer below epidermis. Then its specialization in the structure, that is it has much thickened, that means cell wall is much thickened at corners due to deposition of cellulose, hemicellulose, pectin. Then shape can be either oval or spherical or polygonal in shape. Then they often contain chloroplast, and if chloroplast is present, they assimilate food, that means they can perform photosynthesis. Then cells are tightly packed, no intercellular spaces. They are specialized for providing mechanical support, that is to young growing parts like young stem, petiole of leaf, etc. Sclerenchyma is a dead tissue. It is specialized for providing mechanical support. It is a major mechanical tissue in plants. And it consists of long, narrow cells, thick with a lignified cell wall, and they often have few or numerous pits. And then, based on the variation in form, structure, origin, and development, sclerenchyma is of two types, that is fibers and sclerites. Fibers are thick-walled, that means lignified, elongated, pointed cells, often found in groups. Whereas sclerites are spherical, oval, or cylindrical cells with a highly thickened cell wall, and they have narrow cavities or lumen. And the sclerites are usually found in fruit wall of nuts, then pulp of fruits like guava, pear fruit, sapota, and seed coat of legume and leaves of tea. Complex tissues. Complex tissue consists of different types of cells. Two complex tissues are xylem and phloem. First we will see xylem in detail. The two major functions in which xylem takes part are, first is conduction, second one providing mechanical support. The different components of xylem are tracheids, vessels, xylem parenchyma and xylem fiber. First let us see about tracheids and vessels in detail. Tracheids are elongated tube-like cells with thick lignified cell wall and tapering ends. And lignified, they are dead at maturity. That means they are without protoplasm. And vessels. Vessels are long, cylindrical, tube-like structure. Consists of numerous vessel members. Vessels are made up of numerous vessel members. Each vessel members are with a lignified cell wall and large central cavity. Adjacent vessel members are connected through perforations in their common wall. And these two, that is tracheids and vessels, are the tracheiry elements of xylem. And presence of tracheids and vessel is characteristic of angiosperms. Xylem fiber. Xylem fiber has thick lignified cell wall and they have obliterated central lumen. That is, central cavity is obliterated, indistinct, it's not very clear. And they can be either septate or aseptate, either with a cross wall or without a cross wall. Next about xylem parenchyma. Among the four, that is vessel, tracheid, xylem fiber and xylem parenchyma, the only living component is xylem parenchyma. And xylem parenchyma is mainly involved in storage of starch, fat, tannins. And ray parenchyma involved in radial conduction of water. Then, primary xylem, based on the time of origin, primary xylem is of two types, protoxylem and metaxylem. Protoxylem is the first formed xylem with the smaller vessels. Metaxylem formed later with the bigger vessels. And if the protoxylem is pointed towards the center, then it is known as endark xylem. It is seen in stem. If the protoxylem is pointed towards the periphery, then it is known as exarch xylem, seen in root. Phloem. In angiosperms, the components of phloem are sieve tubes, companion cell, phloem parenchyma and phloem fiber. In gymnosperms, sieve tube is replaced by sieve cell, companion cell is replaced by albuminous cells. Then, sieve tube in detail, sieve tube and companion cells. First about sieve tube. Long tube-like structure arranged longitudinally and are closely associated with companion cells. Then, their end walls are perforated in sieve-like manner to form sieve plate. Mature sieve tube has peripheral cytoplasm, large vacuole, but it lacks nucleus. And it is a living tissue. Then its functions are controlled by the nucleus of companion cells. The next about companion cells. Companion cells are nothing but specialized parenchyma closely associated with sieve tube. Then this sieve tube and companion cells are connected by pit fields present in the common longitudinal walls. Then they help to maintain the pressure gradient in sieve tube, which is necessary for the functioning of sieve tube, that is transport of food material. Phloem parenchyma. Phloem parenchyma consists of elongated tapering cells, and they are cylindrical also. And their cell wall has pits through which plasmodesmata, cytoplasmic connections exist between parenchyma cells, phloem parenchyma cells. Then they are involved in storage of resins, latex, mucilage. Then phloem parenchyma is generally absent in most of the monocots. Then phloem fiber, also known as bast fiber, is nothing but sclerenchymatous fibers, sclerenchyma cells made up of sclerenchyma cells, and they are absent in primary phloem, present in secondary phloem only. And they are elongated and branched, pointed cells with a needle-like apex. And commercially important phloem fibers are obtained from plants like jute, flax, and hemp. Then two types based on the origin, time of origin. Two types of phloem are protophlloem, metaphlloem. Protophlloem first formed with a narrow sieve tube. Metaphlloem formed later with a bigger sieve tube. Tissue systems. Based on the location, tissue systems are of three types. Structure, function are also dependent on the location. The three types are epidermal tissue system, vascular tissue system, and ground tissue system. First about epidermal tissue system. Under epidermal tissue system, the first component, that is epidermis. Epidermis is nothing but the outermost layer of a primary plant body that consists of parenchyma cells, tightly arranged parenchyma cells. It is usually single layered. And cuticle. Cuticle is found outside, that is outer to epidermis. That is present in, it's a waxy coating, present in leaves and stem. Cuticle is absent in roots. It helps to prevent water loss. Then stomata, small opening found mainly in the leaves of, in the epidermis of leaves. Stomata helps in gaseous exchange and transpiration. Then guard cells, a stomatal opening and closing that is actually regulated by specialized epidermal cells known as guard cells. Guard cell contain chloroplast. Guard cells are of two types, that is dumbbell shaped guard cells are found in grasses, monocot. Bean shaped guard cells are found in dicots. Then subsidiary cells, again specialized epidermal cells found in the vicinity of this guard cell or adjacent to guard cell. And these three, that is stomata, guard cells, subsidiary cell, together constitute stomatal apparatus. Then root hairs are unicellular elongations of root epidermis meant for absorption of water and minerals. Then trichome. Trichomes are multicellular. These are stem hairs, and trichomes prevent water loss. Trichomes can be secretory also. It can be either soft or stiff. It can be either branched or unbranched. The next about ground tissue system. All tissues except epidermal and vascular tissues constitute ground tissue system. It is also known as fundamental tissue system. It is usually made up of simple tissues, that is parenchyma, collenchyma, sclerenchyma. It forms the main bulk of the plant body. Ground tissue of leaf is known as mesophyll. Vascular tissues. Vascular tissue system. Composed of xylem and phloem. Based on the arrangement of xylem and phloem, it is of two types, that is conjoined vascular bundle and radial bundle. Conjoined bundle, xylem and phloem are arranged along the same radius. Conjoined bundles are seen in stem leaves. Then radial bundle, here xylem and phloem are arranged on different radii, or otherwise we can say that alternate arrangement of xylem and phloem. This is seen in root. Then based on the presence or absence of cambium, that's a lateral meristem, it is of two types. Vascular bundles are of two types: open bundle and closed bundle. Open bundle means cambium is present between xylem and phloem. Closed bundle means cambium is absent. Then next about the anatomy of plant organs. First about root. Starts with the dicot root. The outermost part is epiblema, nothing but epidermis of root, and it has root hairs. Inner to that, cortex, a multi-layered cortex, consists of thin-walled parenchyma, thin-walled, loosely arranged parenchyma. Then endodermis, the innermost layer of cortex, which consists of, it's a single layer of cells, barrel-shaped, compactly arranged parenchyma. And then this endodermis, radial wall and tangential walls of this endodermis has suberin deposition known as Casparian strips. Suberin is a waxy substance. Then inner to endodermis, pericycle is present, which consists of or made up of thick-walled parenchyma. Lateral roots and vascular cambium initiates in this region. Then pitta. Pitta is the innermost or centermost region, which is highly reduced in dicot root. Then conjunctive tissue, that is nothing but the parenchyma present in between xylem and phloem. Then vascular bundles, number of xylem ranges from 2 to 4, diarch to tetrarch, and vascular bundles radial, exarch. Then stele. Stele means a technical term that includes all tissues inner to endodermis. All tissues inner to endodermis means it starts from pericycle. Then if you see the anatomy of monocot root, which all are the differences? Fundamentally the anatomy is similar because it is root. A few differences are listed here. One is polyarch. They differ in the number of xylem. Polyarch means many xylem, more than 6. Then another difference, pith. Here it is reduced. In monocot it is large and highly developed. Then there is no cambium and no secondary growth. Anatomy of stem. First about dicot stem. Outermost part is epidermis. Epidermis usually has cuticle. Trichomes and stomata, if present, it will be on the epidermis. Then cortex, differentiated into 3 layers: hypodermis, cortical layer, and starch sheath. Hypodermis, just below or inner to epidermis, and it is made up of collenchyma involved in mechanical support. Then cortical layer, inner to hypodermis, it's made up of round, thin-walled parenchyma with the conspicuous intercellular spaces. Then inner to cortical layer, endodermis is seen. Due to the deposition of starch grain, it is known as starch sheath. Then inner to cortex or inner to endodermis, pericycle is present. Pericycle is made up of semilunar patches of sclerenchyma. Then next about medullary rays. Medullary rays are nothing but parenchyma present in between vascular bundles. That is known as medullary rays. Then vascular bundles in dicot stem, ring arrangement of vascular bundle is characteristic of dicot stem. Then other features, conjoined, open and endarch. The next about pith. Pith is well developed in dicot stem. It is well developed. It's made up of round parenchyma with intercellular spaces. The next about monocot stem. The differences. Major difference are hypodermis. Hypodermis is made up of sclerenchyma. Then vascular bundles are scattered in the ground tissue and peripheral bundles are smaller than centrally located vascular bundles. And then each vascular bundle has a bundle sheath, a sclerenchymatous bundle sheath. Then vascular bundles are closed, no cambium, no secondary growth. Then they have large conspicuous ground tissue. See, it is not differentiated into various layers like a cortical layer, endodermis, pericycle and all. Instead, a large ground tissue is present in monocot stem. Then, phloem parenchyma absent. Water containing cavities are present in xylem. These are known as protoxylem cavity or protoxylem lacunae. Anatomy of leaf. First about dicot leaf, dorsiventral leaf. Epidermis. The two epidermis of leaf are upper epidermis and lower epidermis. That is adaxial and abaxial. In case of dicot leaf, numerous stomata are on abaxial epidermis. That is on the lower epidermis. Hypostomatic. Then its mesophyll is differentiated into, mesophyll is made up of parenchyma. It's differentiated into palisade parenchyma and spongy parenchyma. Palisade parenchyma. These cells are first of all adaxially placed, upper epidermis. Then they are elongated cells, vertical and are arranged parallel to each other. Then spongy parenchyma, either round or oval in shape. They are situated below the palisade parenchyma and they extend to the abaxial epidermis, that is lower epidermis. And these cells are with cavities, intercellular space or air cavities are found in between these cells. Then vascular bundle, vascular system. The size of the vascular bundles are dependent on the size of the veins. Size variation can be seen in reticulate venation of dicot leaves. The next about monocot leaf or isobilateral leaf. Here, first of all, stomata equally distributed on both abaxial and adaxial epidermis. That is amphistomatic. Then mesophyll, not differentiated into spongy and palisade parenchyma. Then in monocot, especially in grasses, bulliform cells are present. That is, certain cells of adaxial epidermis, that is upper epidermis, becomes large, empty and colorless to act as bulliform cells. When this bulliform cell becomes turgid, the leaf surface is fully exposed. When bulliform cell becomes flaccid, it makes the leaves to curl. So this is an adaptation to prevent water loss. Then here in monocot leaf, nearly similar size of vascular bundles. That means veins are almost similar in size, seen in parallel venation.
If you are not yet subscribed, please subscribe by pressing the subscribe button below and press the bell button also to get the notifications. If you think this video is useful for NEET preparing students, please share it. Thanks for watching.